We're looking at the respiratory system. First, let's look at the respiratory membrane. So we have type 1 epithelium, which is basically the simple squamous cells of the alveolus. So these are all the simple squamous cells here making this spherical shaped structure. The air comes in to the alveolus. Overall, oxygen will be going towards the blood, and CO2 will be leaving the blood and going into the alveolus. And then the CO2 will go out that way. And so this type 1 epithelium are the squamous cells, which allow for gas exchange. Being a squamous cell, you're very thin, and that allows for gases like oxygen and CO2 to diffuse very, very easily. Type 2 cells are these septal cells, and these septal cells right here release a surfactant, which is important because a surfactant decreases the surface tension due to water within this tissue. Now, if you think about a plastic bag, and if you have a film of water within that plastic bag, you know that the walls of the plastic bag stick together when there's a film of water inside of it. But if it's dry, it's easy to open up the plastic bag. So the alveolus works in a very similar way. If you have a film of water on this side and then another film of water on the other side, water is sticky to itself, so the two layers of water can attract each other and stick together, and that will cause the alveolus to collapse. And so you need a cell, such as this type 2 cell, which releases a type of oily surfactant that interrupts these water interactions. And that's what those are useful for. And then the third type of cell that you have there that's important is the macrophage, which in parentheses here is nicknamed a dust cell because every time you breathe in, there's a lot of particles in that air that you've breathed in, such as dust, and we need some sort of vacuum cleaner type cell that can go and move around like a Roomba, basically, move around and phagocytose or clean up all the solid particulates that are found in air. Here we're going to look at the blood supply to the lungs, and we see we have pulmonary arteries, which are blue, so just realize that the colors here are reversed then from systemic circulation, and the pulmonary veins, which are red. And so the way this works is that this blood is coming in from the heart, and it's coming in and it's blue blood because it has low oxygen levels, so it's coming in. It goes towards a capillary bed, which is right here. And in this capillary bed, it'll go from blue to purple as it gets oxygenated, and then it will leave as red blood, and then it'll make its way to the venule, and from this venule, it will go back towards the heart. And then after that, the heart will pump it to the systemic circulation so it can reach the tissues throughout the body. We need to talk about pressure because airflow is governed by the same principles as liquid, such as blood. We talked about blood and pressure and flow there. So a lot of these concepts are going to be the same, but we're going to talk about these concepts regarding air. Now, the first thing to notice is that if you are right here in the middle of North America, let's say, you have a column of air from the atmosphere that's sitting on top of you, and that column of air is putting a certain pressure on your head, and that pressure has been measured to be 760 millimeters of mercury at sea level. So at sea level, New Orleans is at sea level. At sea level, 760 millimeters of mercury. How did we do this? We took, in the early days, that is, we took basically a cup of mercury. We stuck a cylinder inside that cup, and the mercury would climb up the cylinder, and then we would put a ruler next to that cylinder and measure how many millimeters of mercury were in the cylinder. So basically, the atmospheric pressure was pushing the mercury down, and that mercury was basically being forced to go through that cylinder. So that ends up being 760 at sea level, or sometimes we'll just say 1 atmosphere, right? 1 atmosphere is the same as 760 millimeters of mercury. And so just realize that if you're down here, right, at lower elevation, at sea level, you have a larger column of air. But then if you climb up the mountain, right, and you go up to the mountain, then on the mountain you have a smaller column of air. And so the atmospheric pressure at a higher elevation is going to be lower. For example, here in this picture, it shows that it's 714 millimeters of mercury. The higher you go, the less atmosphere there is. The less atmosphere, the less that pressure will be felt. Now, this is important because that pressure is going to drive gas exchange at the alveoli. So at a higher pressure, it'll be easier for oxygen to be pushed into your body. At a lower pressure, it'll be less easy to push the oxygen into your body, which would make breathing more difficult at higher elevations. Let's talk about the respiratory muscles. So in general, you have inspiratory muscles and basically two types of breathing. The first one we're going to talk about is quiet breathing. So when you inspire, when you breathe in, for example, your sternocleidomastoid pulls these bones, the sternum and the clavicles upwards, increasing the size of the thoracic cavity up here. The diaphragm is another example. Will flatten out and will move downwards, increasing the size of the thoracic cavity down there. And then you have the intercostals, which will open up the thoracic cavity to the side. And so when these muscles contract, basically you get an enlargement of the thoracic cavity, decreasing the pressure inside the thoracic cavity, forcing air to flow into the thoracic cavity. And so those are going to be your inspiratory muscles. Now, during quiet expiration, when those muscles contract, everything goes back to its regular size and air is basically pushed outwards. So they contract during inspiration, they relax during normal quiet expiration. However, sometimes you have a fight or flight response. A sympathetic response requires more forceful breathing, and so we call that forced expiration. In this case, you're going to activate many more muscles, and those muscles will be working more powerfully. And so now, in order to push that air out faster, you have forced expiration. And so, for example, your diaphragm will start activating in order to go back up to force the air out. Your rectus abdominis, right, which is your abdominal musculature, will start working harder from the abdominal cavity to push on the thoracic cavity to force that air out. And so now we have this sympathetic type of breathing or forced breathing on the other side. So at rest, it's going to be quiet, normal breathing, but during a fight or flight sympathetic episode, it'll be forced expiration. Let's look at control of respiration using the nervous system to control it. And so really it starts with the brainstem here. what we're looking at is the medulla oblongata and the pons. Those are the two areas that we want to look at. So let's start with the medulla oblongata. First of all, you have what is called a pre-Botzinger complex. And the pre-Botzinger complex is basically a pacemaker that gives you a rhythm. And it gives you a rhythm, so think about respiration rate, how many breaths per minute you are doing. That's what the pre-Botzinger complex does. So it's kind of a pacemaker, but it's located in the central nervous system. Also, what you have is a group of neurons that is important for orchestrating the quiet breathing. And if you remember the quiet breathing, you have to contract the breathing muscles during inspiration, but you just relax them during expiration. So that's a type of breathing that has to be orchestrated by the central nervous system. However, if you have a fight or flight response and you go into sympathetic mode, then it switches gears and it goes to the ventral respiratory group. And in the ventral respiratory group, it starts doing that forced breathing, where basically both inspiration and expiration are going to use muscle contractions. Breathing rate will increase, breathing strength will increase. Also, further up in the pons, you have what is called a pontine respiratory group. And this pontine respiratory group is important for coordinating other activities with your breathing. So, for example, chewing or moving. Think about yourself eating a sandwich at lunch, talking to a friend at the same time. All of that is very complicated. And when you're eating and talking, and you also have to oxygenate your blood at the same time, and to do it properly without, for example, breathing in while trying to swallow your food simultaneously, you need a mechanism to do that for you in your central nervous system where you don't have to think about it, but it's doing it for you. Other factors that affect this. So, for example, cortical influences. This just means that the cerebral cortex is influencing the brainstem. Okay, what do we mean by this? These are your thoughts. And so, if you're relaxed and you're not thinking about anything stressful, your respiration rate will decrease. However, if you are stressed out, if you're watching a scary movie, if you're scared, then those thoughts will influence the brainstem, and by influencing the brainstem, they will increase respiration rate. Clinically, we always have to think about the pH levels of the blood, and this is where the chemoreceptors come in. Chemoreceptors are monitoring H+ levels, CO2 levels, O2 levels. Where are they located? So you have the aortic bodies in the aorta, so the blood is getting pushed out the left ventricle through the aorta. It's going to run into these clusters of neurons there, which are going to monitor the pH of that blood. Also, as the blood goes up through the carotid arteries, it's going to run into the carotid bodies, which are also going to monitor the pH levels. So imagine a situation where your partial pressure, that's what that P stands for, your your PCO2, the partial pressure of carbon dioxide goes up. That means that you need to get rid of it. So how are you going to get rid of it? Well, the chemoreceptors that are peripherally found in the aorta or in the carotids are going to realize this. You do have chemoreceptors centrally located in the brainstem as well, in the medulla. They're going to send nerve impulses to those respiratory groups in the medulla, and those are going to cause changes in the effectors, which in this case are the muscles of the thoracic cavity. And so if your CO2 levels are too high, you need to hyperventilate. You need to increase respiration rate so that you can drop your CO2 levels and therefore drop your acid levels and bring your pH back to normal. Okay? So this is a classic negative feedback loop using chemoreceptors. Other factors affecting pulmonary ventilation have to do with the tissues. So first of all, let's look at the alveoli, which in a healthy individual are nice and round shaped, kind of look like grapes, and those connective tissues full of those elastic fibers are able to give elastic recoil, which just means that once the air comes in and bounces off the walls of that alveolus, it'll kind of expand a little bit and then push back, and that pushback is gonna help to get that air back out, helping with ventilation. So individuals who have smoked for many years can have emphysema, and emphysema is where the connective tissues start breaking down, and you can see that the shape of the alveoli are now no longer those nice round shapes, and that's going to cause that elastic recoil to go down. Also, the lungs get stiffer, and so this decreases the compliance of the lung, or the ease with which the lungs and the thoracic wall can expand. And this will also, of course, affect the breathing. Another clinical case is asthma, which is when somebody has an allergic reaction and releases too much histamine, and that histamine basically causes this inflammation that takes place within the bronchiole. So here's a normal bronchiole, nice and open, with a large radius. And when you have a large radius, that means that your resistance is low, your flow is high. The same thing that we saw with blood vessels applies to air flow. But with asthma, this inflammation causes the lumen of this tube to become very small and constricted, and now the flow is severely reduced, causing the person to have a very difficult time breathing. And so antihistamines, of course, will help with this, but if you want something more potent, you would just use epinephrine, and the question is why? Well, epinephrine is a fight or flight hormone, right? It activates your sympathetic nervous system. During a sympathetic nervous system response, if you have to run away, your muscles are working hard to run away. That means they need oxygen, which means those airways are forced open in order to get that flow, to get that oxygen to those muscles. So epinephrine is a faster, more powerful way to pop open those bronchioles. Let's look at some breathing patterns. This right here is normal respiration, which has a tidal volume that's normal, that's basically the same going in and out. It's about 500 mLs. You have the tidal volume going in and the tidal volume going out, in and out. Okay, it pretty much stays the same. When you have neurological damage, right? So imagine that the brainstem gets damaged in some way and those nuclei get damaged. You go into periods of apnea where you're not breathing. And then because you're in apnea, your CO2 levels go up because you're not getting rid of them. You're not blowing off CO2 if you're not breathing. And then you basically go into acidosis because of it. And now your the mechanism for homeostasis to fix your pH levels kicks in, and you go into basically hyperventilation. And so you can see that these curves are very elevated, and then it goes back to apnea. Drugs can also do this, and so you see a very similar pattern down here with apnea followed by hyperventilation and then apnea again. We have the first one that's known as Biot's respiration, and then one that's very similar to it known as Cheney-Stokes. One that's very common in diabetics is Kussmaul breathing. And the reason why this is common in diabetics, this is a type of metabolic acidosis where glucose is not able to get metabolized, and so the individual starts metabolizing fats, which leads to ketones. Now, ketones are acidic, and so you get this acidosis from the metabolite of the ketone, and because of that, the lungs try to fix that acidosis by blowing off CO2, by getting rid of CO2. And so what you see is this hyperventilation that just keeps going until that acidosis is fixed. Now, before we get to gas exchange, we have to talk about laws that basically govern how gases work. The first one we're going to talk about is Boyle's Law. In Boyle's Law, what we say is that we have this container, and this container holds a certain amount of gas in it, being depicted by these orange dots. And basically those dots are going to collide into each other, bounce off of each other, and then bounce on the walls of this container. And the pressure gauge here is reading a number 1 on the pressure gauge. And 1 is just an easy number to use to depict this. So basically the volume we can say is 1 liter, the pressure is one atmosphere, let's say, and then we want to know what happens when we decrease the size of the container. So we use a piston and we push the piston down, and so we decrease the size of the container and we cut it down to half, so the volume becomes 1/2 of a liter. And now we look at the pressure gauge and it says 2 atmospheres. And so basically by decreasing the container by half, we have increased the pressure twofold. And so what you need to think about in this case is your thoracic cavity. Your thoracic cavity, which has the lungs inside of it, that thoracic cavity will expand during inspiration and will then get smaller during expiration. That's exactly how basically the air is either forced into the lungs or out of the lungs, is using Boyle's law. The other way to think about this is you have 2 rooms, area A and area B. They each have the same amount of gas inside of them. They're the same size. They're connected by a corridor. You increase the size of B, you expand it, all of a sudden the pressure decreases in B, and the gas is going to flow from a higher pressure in A to a lower pressure in B. You can do a similar experiment where you decrease the size of the B chamber, putting more pressure in chamber B, forcing the air to flow from higher pressure B to a lower pressure A. Two more laws that we have to look at. One is called Dalton's law, which basically states that each gas exerts its own pressure on the overall pressure. So the overall pressure at sea level of the atmosphere is 760 millimeters of mercury, right? That's the overall pressure. However, the atmosphere is composed of mostly nitrogen, by the way, which is kind of surprising for us because we don't really talk about nitrogen too much. It doesn't serve as much of a purpose for metabolism as oxygen and carbon dioxide do. But notice that 78% of the atmosphere is nitrogen. All right, so let's ask, well, how much pressure does nitrogen put just by itself? So we take that 78%, we put it in decimal form, we multiply it by the overall, 760, and we get 597.4 millimeters of mercury. So that's how much contribution nitrogen makes for the overall. Oxygen is 20%. So oxygen, you take the decimal 20%, multiply it by 760, you get this 158.8 millimeters of mercury, and that's how much oxygen contributes. And CO2, surprisingly, is very, very small. So CO2 at 0.04%, you multiply that decimal by 760, and you get 0.3 millimeters of mercury. So a very small percentage from carbon dioxide. The second law that's important is Henry's law. The second law on this slide that's important is Henry's law. And Henry's law states that that partial pressure, right, so the P in front of all these gases stands for the partial pressure. The partial pressure of the gas will influence how that gas dissolves in a liquid, right? And so if the partial pressure is higher, it's going to push that gas into the liquid and cause it to go into the solution of the liquid. So whether it's a pond of water, or in our case, because we're doing human anatomy and physiology, we're talking about blood, the partial pressures will drive how those gases dissolve within the bloodstream. A great example of this and how it can go wrong is known as the bends. Called the bends because when a diver, let's say, rises up too quickly from a column of water, the pain will be so excruciating that the diver will kind of be hunched over, bent over in pain. So at sea level, we know that the pressure from that column of air is 1 atmosphere, so at sea level, 1 atmosphere. When you dive into that column of water, now not only do you have the column of air on you, you also have the column of water. If you dive down 10 meters, it's about 2 atmospheres worth of pressure. What that's going to do to the nitrogen, which if you remember, nitrogen was the most, the gas that was in highest quantity, what's going to happen is that nitrogen is going to get pushed into solution into your bloodstream. And so now that nitrogen is in solution in the bloodstream because of that high pressure. If you rise up too quickly, the pressure quickly drops, and all of a sudden that nitrogen is not getting pushed into solution anymore. And so what happens is it starts bubbling out of solution. And so where does it go? Now you got these bubbles, these nitrogen bubbles that are bubbling out of solution. They can go inside your joints, and that's what's causing you all that pain. They can go, they're going to be in your veins and in your arteries. They can even go into your nervous system and can cause strokes and seizures. So what we do is, to fix the problem, we put you in a hyperbaric chamber, which is basically a container that increases the pressure, pushing the nitrogen bubbles back into solution, basically. And then we slowly release the pressure so they can bubble out more slowly. Because the thing is, if they bubble out slowly, it's not so bad. It's when they bubble out too fast that is the problem. So let's see how these pressures also drive the gas exchange that we need to do over here. There are 3 ways in which carbon dioxide gets transported inside the blood. 70% of it is from carbonic acid. And just to remind you, this is when carbon dioxide and water combine to make carbonic acid, H2CO3, which splits into acid H+, and bicarb HCO3-. So basically we're taking the CO2, which is in a gas state, and masking it to make it soluble inside the blood through this carbonic acid. 23% is from carbamino compounds, amino for amino acids. So we're talking about CO2 binding to proteins, specifically the most important one being hemoglobin, which takes the CO2 and binds it. So hemoglobin, famous for carrying oxygen, also carries carbon dioxide. 7% of the carbon dioxide stays as a gas dissolved inside the plasma. So 7% of it just goes into the plasma and stays as a gas. So here's the picture of it. Now, right here on the left, what we have are the respiring tissue cells. So these cells are doing respiration. That means they're making a lot of CO2, and that CO2 has to be picked up by the capillary, which is coming in with the red blood cell. So let's check out the percentages and see if they match. So 7% dissolved CO2 gas in the plasma. 23%, it diffuses from higher concentration to lower concentration, and it goes to this HB, which stands for hemoglobin, giving us our hemoglobin and carbon dioxide attached. The hemoglobin is carrying the carbon dioxide. And 70% is where the CO2 basically fuses with water with the help of this enzyme Ca, which is carbonic anhydrase. And basically this enzyme helps the reaction to go much faster, giving us carbonic acid H2CO3. H2CO3 splits into the acid H+, and into the bicarb HCO3-. Let's look at what happens with the H+, because obviously you don't want a lot of H+ accumulating inside a red blood cell, because H+ leads to acid. So what we're going to do is we're going to neutralize it, and hemoglobin will actually neutralize it. So hemoglobin will grab that H+ and neutralize it. Hemoglobin showing us that it has many functions besides just the transport of oxygen. What about the bicarb? So realize that the bicarb has to get pushed out of the RBC because it is known as the plasma buffer. So we want the HCO3- to get basically pushed out of the RBC and go into the plasma to act as a buffer. The way that that works is that if there's an accumulation of acid H plus inside that blood for whatever reason, the HCO3 can grab it and neutralize it. So it'll grab it, it'll neutralize it, and it'll give us the H2CO3 back again, which now is no longer acidic. Now, why do we have this chloride shift over here? Notice that bicarb is negatively charged, which means that if we keep throwing out something negative, we need to replace that outward flow of negative with an inward flow of something negative. And so what we do is we basically exchange chloride, which is negatively charged, for the bicarb, and that's known as the chloride shift. What this does is it maintains the polarity of the cell. Remember that from the nervous system and the muscular system, we have depolarizations and repolarizations, which use ions, which are positively or negatively charged entities, which either flow in or out of a cell, changing the polarity. We don't want to do that in this case, and so we do this exchange called the chloride shift. What about oxygen? Well, oxygen, easily enough, what we're going to do is we're going to basically take the oxygen that's bound to hemoglobin, we're going to disassociate that oxygen and let it, basically the hemoglobin will disassociate from it, and it'll let the oxygen flow out of the blood to the respiring tissues that of course need that oxygen to make ATP. Now, this RBC is going to have to cruise all the way to the lungs, and now we have to see what happens at the alveoli. So at the alveoli, this same red blood cell arrives, and we basically have to reverse the whole thing. So notice that the oxygen from the alveoli, in this case, is going to go into the RBC, will attach to the hemoglobin and make a oxyhemoglobin molecule for us. 98.5% is behaving like that. 1.5% is just staying dissolved as a gas. What about CO2? Well, with CO2, we have to reverse everything. So we need to take the bicarb, HCO3 minus, which was in the plasma, we need to bring it back into the RBC, and we need to do another chloride shift or a reversal of the chloride shift where we get that chloride that we brought in and we put it back out into the plasma, so we can do it all over again when we go back to the tissues again. So now that bicarb is inside the RBC, it's going to find the H plus that was attached to the hemoglobin, and it's going to grab that H plus, it's going to combine with it to give us our carbonic acid. And carbonic anhydrase is going to reverse this for us. This equation can go forwards and backwards. In this case, it's going backwards. And so that carbonic anhydrase is going to push this equation to the left, giving us our carbon dioxide back and our water back. The CO2 simply goes from higher concentration to lower concentration, or higher pressure to lower pressure, I should say, and is going to get pushed out to the alveoli. The hemoglobin will also detach from the CO2, and again, from higher pressure to lower pressure, the CO2 will get pushed out. Remember your percentages, 70%, 23%, and then the dissolved gas also will just bubble out into the alveoli, and then all this CO2 can go out of the alveoli into the bronchioles and into the bronchi, and that's how you breathe it out. Now, hemoglobin behaves in very interesting ways in order to be able to oxygenate the tissues that need the oxygen more than the tissues that don't need the oxygen. So certain tissues are working harder than others at any given time. That means they're doing more metabolism, more respiration. And what you want is a situation where the hemoglobin will donate the oxygen that it has to the tissues that need it the most. Now, when a tissue is doing respiration, that means its CO2 levels are going up, and therefore the hemoglobin that arrives to those tissues will be much more generous with the oxygen and will give the oxygen to the tissues that are showing higher CO2 levels. Because the idea is if you have higher CO2 levels, you're doing more metabolism, you're doing more respiration, that means you need more oxygen. Also, because CO2 levels are high, what we're going to see is lower pH levels, and so higher H plus levels. And so again, if the pH levels are low, again, that hemoglobin will be much more generous to those tissues. So this effect is called the Bohr effect. And so basically, hemoglobin behaves differently with the tissues that need it the most. It's much more generous with its oxygen. It likes to donate the oxygen to those tissues. The other thing that RBCs do is that RBCs, when there is a lot of need, when the body is undergoing a lot of metabolism, the RBCs will themselves generate a molecule known as biphosphoglycerate, or BPG. And this BPG is a trigger to unload the oxygen. So imagine that your body is in overdrive, you're doing a lot of work. The RBCs realize this. They make this BPG, which is a molecule that serves as a trigger to make the hemoglobin much more generous. So it'll start donating much more oxygen to the tissues so that those tissues can get oxygenated. A great example of this is when you're fighting off an infection and your fever goes up and your body temperature going up, your fever is basically a mechanism to fight off the infection better. And one thing that it does is it gets the hemoglobin to be much more generous, so these oxygens start getting released from the hemoglobin and they start going towards the immune system, which is going to use that oxygen for energy in order to fight off the infection. Another thing that we saw with hemoglobin is that hemoglobin, of course, carries oxygen, but it also carries CO2. And so the Haldane effect basically says that when the hemoglobin has low levels of oxygen, right, so it's not really saturated with oxygen, then it is much stickier to CO2, and CO2 likes to bind to that hemoglobin better, which makes total sense because if the hemoglobin is unloading the oxygen at a respiring tissue, that also means there must be a lot of CO2 that needs to be grabbed from that tissue and brought back to the alveoli, and that's known as the Haldane effect. So we're going to look at some curves that explain a lot of this, and these are called oxygen saturation curves. So what you have on the x-axis is the partial pressure of oxygen going from 0 to 100, and on the y-axis you have the saturation of the hemoglobin going from 0 to 100. The way to think about this is that you have a hemoglobin which has 4 binding sites for oxygen, and if the hemoglobin is fully filled with oxygen, all 4 binding sites are filled with oxygen, then it is 100% saturated. And so we would be looking at this value right here. However, if that hemoglobin becomes generous and donates one of these oxygens and gives it to a respiring tissue, let's say, then all of a sudden now you're looking at 75% saturation. You have 3 out of 4 of those binding sites filled. Now, if that hemoglobin gives off another oxygen, let's say, and gives it to a respiring tissue, now you're looking at 50% saturation. What about the partial pressure? Well, the partial pressure comes from the conversation we had about gas exchange. And we saw that the partial pressure of oxygen in red blood, oxygenated blood, was 100 millimeters of mercury. And in blue blood, in venous blood, deoxygenated blood, it was 40 millimeters of mercury. And that's why we're looking at these two values very, very specifically and very, very carefully. And so notice that when the partial pressure in that red blood is at 100, well, no surprise, the hemoglobins are fully saturated because that is, after all, oxygenated blood. So you have oxygen bound in all four slots on each hemoglobin molecule, giving you 100% saturation. When, on the other hand, you release that oxygen and now you're in those blue vessels, you're at 40 millimeters of mercury of partial pressure of oxygen. And when you look at this value, you notice that the hemoglobins now are at 75% saturation. So if you were to draw them, this hemoglobin would have oxygen in four spots at 100%, but this one right here would have it in three spots at 75%. So this is kind of cool because it makes you realize that blue blood is not actually deoxygenated fully, is it? Right? So red blood is 100% saturated. Blue blood, on the other hand, is at 75% saturation. So it's still pretty oxygenated. That's why you can hold your breath for a few minutes, let's say, and not die, is because you still have oxygen cruising through the blood, and that oxygen is still able to go to tissues that need it and keep the metabolism going temporarily. Now, what does happen is once that partial pressure goes below that number 40, what we see is a nosedive effect. You can see the slope of this curve is very steep after that 40, basically bringing you down very, very quickly. So notice 75% at 40 partial pressure, but look at 20 partial pressure. 20 partial pressure, you're looking at 35% saturation. So you've lost a lot of oxygen if that partial pressure of oxygen drops too low. So what's cool about this is that as long as you're hanging out in this zone right here, which is, normal physiological zone, then you should always be pretty oxygenated, right? Your arterial blood should be at 100 pretty much percent saturation, and your venous blue blood should be at about 75. So you're pretty oxygenated. It's only when you dip below those values that you start seeing problems with oxygen levels in the blood. Let's look at the Bohr effect in detail. Now imagine a tissue that is doing a lot of respiration, has very high metabolism. What's going to be happening in that tissue is that the CO2 levels will be very high, which means that the H plus levels will also be very high, which also means that the pH levels will be low. So with a tissue like that, think about a muscle that's working really hard, a skeletal muscle. You're working out or you're running away from something. That muscle will be doing a lot of metabolism, and therefore these will be the values that we'll see in that tissue when that blood approaches. So that blood is going to have hemoglobin in it, and that hemoglobin is going to have oxygen attached to it. And what you want for that specific tissue is that you want the hemoglobin to be extremely generous to that tissue. You don't want it to hold on to the oxygen. You want it to basically release the oxygen and give it to the tissue that needs it. And that's exactly what we see, is that the hemoglobin does change how it behaves based on which tissue it runs into. And so this black line right here is our original curve, and that's your standard oxygen saturation curve. But notice that the red line down here is showing us, well, what happens when the blood pH drops very low at 7.2, is that at every single point on that red curve, the hemoglobin is less saturated, right? It's going to have less oxygen on it. The reason why it has less oxygen is because it is giving the oxygen to the tissue that has that very low pH. Now, what we say is that we have shifted the curve to the right. And that's because you can kind of draw an arrow, and that arrow shows you that you have shifted the curve to the right. The opposite is also true. So if you run into a tissue that's not doing a lot of respiration, you will expect that the CO2 levels will be low, therefore the H plus levels will also be low, and the pH levels will be high. And in fact, when the hemoglobin goes to those tissues that are not doing very much, what we see is that the curve gets shifted to the left. So we shift the curve to the left for those tissues. And so at every single point, the hemoglobin, right, on that blue curve is always much more saturated with oxygen than the black curve, and definitely much more saturated at any given point compared to the red curve. And we've shifted it to the left in that case. So that's the case for pH levels, and that's also the case for CO2 levels. So again, the hemoglobin responds to the CO2 levels. If the tissue is not doing a lot of metabolism, you will expect high, you will expect low CO2 levels. And so what we're going to do is we're going to shift the curve to the left, and the hemoglobin will be very greedy. It's not going to give the oxygen away very easily. However, when we compare it to that red curve, where we have very high CO2 levels, well, that just means that that tissue is doing a lot of work. And so at that point, that hemoglobin, because it is so generous, right, that the amount of oxygen it has on it will not be as high, right? Instead of being 100%, it'll be like 70% or so, right? Let's compare these two at the same point, right? You see this difference there, and that's because that oxygen has gone away from the blood, away from the hemoglobin, and towards the tissue that needs them, and that's known as the Bohr effect. A continuation of this concept has to do with temperature, because after all, if you think about a muscle that's doing a lot of work, a lot of metabolism, then that muscle, with that metabolism being so high, the temperature will go up. And when the temperature goes up, the hemoglobin also responds to those changes in temperature. And so here we see that this is the regular black curve that we started with. When we have a very elevated temperature down here, we shift the curve to the right, and basically our hemoglobin is very generous. At any given point, we'll have less oxygen on that hemoglobin, right? Less saturation at any point compared to the black curve. But then when the tissue is not doing a lot of respiration, we can expect the temperature, right, to be low. And when the temperature is low, then the hemoglobin is very greedy with oxygen. It holds on to oxygen, and the saturation of that hemoglobin for those tissues is always much, much higher because of that. The last associated concept with this conversation has to do with the fact that fetal hemoglobin is much stickier to oxygen than maternal. And so what we've realized is that in the fetal period, the hemoglobin that you express is a slightly different structure than when you're an adult. And so if you think about the placenta being the interface of gas exchange, where you have the mother's blood on one side and the fetal blood on the other side, the two bloods don't mix, but you do need to exchange stuff. And so the question is, for oxygen, well, what happens? Well, if the hemoglobin on the fetal side is much greedier, it's going to attract the oxygen. And if the mother's hemoglobin is much more generous, right, then it won't hold on to the oxygen. It will allow for the oxygen. It will allow for the oxygen to move from her bloodstream to the fetus's bloodstream, which is what you want. And so now we're oxygenating the fetal blood, and now that fetal blood can go and cruise to that developing body, which needs so much energy to grow and develop. And of course, we wouldn't have it any other way. And the last thing we're going to look at is carbon monoxide poisoning. Carbon monoxide CO instead of CO2, right? So carbon dioxide is CO2, carbon monoxide is just CO, is a gas that we get in especially high quantities during combustion or during burning stuff. And so, for example, cigarette smoke, engine exhaust, fumes from furnaces release a lot of carbon monoxide. And when this carbon monoxide is in very high quantities, it goes into the bloodstream and it binds to the hemoglobin. So imagine you've got this hemoglobin inside your bloodstream, and this combustion is causing all of this carbon monoxide to come out and go into the blood. Now it attaches to the hemoglobin, and it basically attaches much more strongly than oxygen. So notice it binds 210 times more tightly than oxygen does, and so oxygen doesn't really have a chance. So when oxygen, let's say, is going to try to bind to it, it can't, because all of the sites are taken up by carbon monoxide. Okay, so of course, then carbon monoxide, which is useless for metabolism, once it goes to the respiring tissues can't use it, and so basically they're starving of energy. They cannot make ATP, and you die as a result.